So I chose today to talk to you about sex in insects. And this is a project that has been ongoing in the lab for a little while, but it's a project really in the building. So I wouldn't be showing a lot of finalized data, but at least I'll show you where things would be going with this project. So this is about coevolution. And really, for us, what we are interested in is to try to understand how the sexes, which share the same genome, get to diverge phenotypically, sometimes dramatically, and how, when one sex diverges, how does that affect the other sex? So this is the sense of my title, where we see trying to go towards this understanding how this sexually antagonistic evolution drives these patterns of escalation of armaments and arms race that David just introduced earlier. So people who have been contributing to this work, most importantly, David here, who's a postdoctoral fellow and he's a bioinformatician, a few interns, master students, also Séverine, who is a lab manager, and she has a lot of expertise in these animals. And also some colleagues, like a lot of the work I'm going to show is based on work done by Joran Arnquist and Locke Rowe, who established these models for ecology and evolution, and somehow from Adel Husby and from Felipe Moreira. So the talk today is about sexual conflict. So what is sexual conflict? Is every time the two sexes diverge in terms of their evolutionary interest, that creates a conflict, which is more or less all the time and in all lineages. So how does it work? So if you look at the various fitness components here, survivorship, fecundity, and mating success, you can have situations like this classic example of peacocks and peahens, where you have an extravagant phenotype, which is the tail, that is constructed by the same genetic basis in both sexes. But this phenotype would come with some cost in terms of survivorship for the males, but this cost will be largely offset by benefits of mating success. If the female would have to carry the same phenotype, and that would make sense because she shares the same genetic basis of this phenotype, the female would have to pay the cost without any benefit. And this is because males, or this is a mating system where males are trying to convince females to mate, and that's how they achieve their fitness. So when the net fitness of this kind of phenotype is positive, then the phenotype is going to be favored and it will spread. There's a lot of theory and predictions about these patterns, and there's also a lot of understanding of how it works at the phenotypic level. Where things are a bit difficult is to understand the genetic basis of these patterns. There are a few examples. This is one of my favorite examples in the salmon, where you have the two sexes that disagree on body size and age at maturity. So females are favored if they mature later and they are mature at larger body size, whereas males are favored if they mature earlier with a smaller body size. So this creates a conflict over this trait. And in this paper, what the authors have shown by using GWAS analysis is that there's a gene, a large effect locus, where the gene exists in two alleles in the population, and one of the alleles is going to be dominant depending on the sex it will find itself in. So they call this sex-dependent dominance. So the dominant allele in males is recessive in females, and vice versa. So these kind of examples are not very common, and this is one thing we are trying to accomplish in our lab. So we're doing this by focusing on these nice animals called water striders, who I'm pretty certain all of you have seen them in ponds and rivers. So we are focusing on a type of conflict this time, which is a conflict over mating rate. So as you know, males and females have to interact, and they have to mate in order to reproduce, but they disagree on the frequency of this mating rate. So if you look at this graph here, So this has been shown with empirical data. Females are going to achieve maximum fitness with an optimum of mating rate that is much lower than that of the males. So females are going to be favored if they are able to keep the mating rate at the optimum that gives them maximum fitness, whereas males are going to be favored if they mate more and more frequently. And you can think about this because of asymmetry in investment in gametes, and because of differences of cost of mating for both sexes, being for females it's costly to mate more than a few times. So how does this work? So when this happens, basically males are going to be selected if they stay close to the optimum of mating rate, and females are going to be selected if they stay closer to the lower optimum. This generates very recognizable interactions where you have in this kind of interaction, it's called pre-mating struggle. The male is trying to grab the female and force a mating, and the female is going to respond with a very vigorous and most of the time quite successful struggle to try to dislodge the male and get rid of the male. So what happens here is that as time goes, males that mate more are going to be to spread, and those are usually males that change in a way that allows them to mate more. And females that mate less are going to be favored, and those are females that also evolve phenotypes that allow them to mate less. So these phenotypes are called sexually antagonistic phenotypes, and there are many, many examples, and especially in water stratas, these are very, very common. And here I'm showing a few examples, like for example, this antenna turned into or elaborated into grasping structure. You have these kind of structures in the abdomen. In females, you have this kind of spines and this crazy type of spines. And all of these phenotypes in males are directed towards allowing the male to grasp, and in females are directed towards allowing the females to reject. And sometimes this ends into an arms race where the sexes change in a correlated way. And what you see here, for example, this is the female and the male of the same species. So females have these erect spines on the abdomen that allows them to block the male when the male tries to mate, and therefore it allows them to stay close to the optimum of mating rate because it allows them to reduce mating rate. So males changed in a way where they have this shape of the abdomen which renders the spine inefficient in the female. So these are very common correlated patterns of changes between males and females, and these are the things we are trying to understand, how these patterns of coevolution happen from a genetic perspective. We've done throughout the years a lot of work showing how some very important developmental genes can actually construct these changes during development. So here in this example, for example, you have males with these elaborated antennae that have a very specific function. They function like a key and a lock to grab the female and to force a mating. And if you inactivate a gene called distalis, so all these phenotypes you see disappear, males will have now antennae that almost look like female antennae, and they also fail in their interaction with the females. They can't grab the female and they can't mate. So we've shown this in many, many examples. You take any example, you can actually predict or you can guess which developmental gene is going to be involved. And here I'm showing you, for example, these two called male abdominal processes. If you knock down a gene called abdominal B, they're lost or double sex. Here, these are third legs of males that are also claspers. If you knock down a gene called UBX, then they're all left. So we know that developmental genes can be very important, but during the development of the trait. You can go even a step further and you can look at a phylogeny in a lineage where you have many independent instances of elaboration of the antennae. And each time we knock down distalis, we lose this elaboration. So it seems like the same pathways or same genetic networks are being mobilized independently for this. This is also true for the third leg. So you can see here all these examples are independent instances where the males have elaborated third legs. And each time when we knock down the gene UBX, we lose these elaborations, regardless of the phylogenetic position or the shape of the elaborations. But the big question that remains is, are these genes or these loci that I'm talking about, distal S, double sex, abdominal B, are these loci undergoing conflict themselves? So what are loci that are undergoing conflict? So those are haplotypes or alleles that when they occur in one sex, or we give an advantage in this process to one sex, that comes to at a cost to the other sex. So these are called sexually antagonistic loci, or loci that are undergoing conflict. And to know which ones are those, we need to find the alleles and the haplotypes that are involved in these traits, and that when they are in one sex, they give advantage to that sex, and they are costly to the other. And so the experiment I've showed you so far does not tell us any of this. So far what we have been doing, basically, we have gene networks. We have each of these genes, part of this network, maybe central, maybe peripheral. And what we have been doing, we're just blocking them, which basically disrupts the function of the entire network, and we lose the phenotype, which tells us that these genes are contributing to this phenotype during development, but doesn't tell us about their role during evolution of these phenotypes. So how do we want to do this? To do this, what we need systems where we can actually do some mapping and try to associate phenotypes with allelic variants in natural populations. So the way we want to do it, we want to have, if you take, for example, these three species, you have this one with this derived state, which has this sexually antagonistic trait called male abdominal processes. And what we are trying to do is to compare these species here and to figure out what kind of alleles and what kind of genes are responsible for these traits that distinguishes this species from the rest. And are these alleles part of the ones we've shown that are important during development or not? That's a question we still don't have an answer to. And to reconstruct the escalation, now we need to bring also the other sex. And we need to understand, or try to understand, when one sex changes, which alleles are involved, and when the other sex responds, which alleles are involved, and how these patterns of escalations, adaptation and counter-adaptation happen, but at the genetic level. So because of this, there are a bunch of questions that are still open, and we would like to be able to tackle them. So for example, how does the adaptation and the counter-adaptation in each of the sexes happen at the genetic level? What are the genomic targets of selection? Are these traits complex, or are they simple traits? And this is important because most of these traits are known to evolve in really short time frames. So we don't know whether there are just a few mutations that are enough for the trait to evolve, or many more. And how does the escalation operate? So why has this been difficult so far? This has been mostly difficult because the field has a sort of dichotomy, or there's a mismatch between, for example, systems where we have powerful genetics, like Drosophila, and systems where we have a deep understanding of how this escalation of armaments happen, and where there are conspicuous phenotypes that we can actually measure. So water striders have been very good models for this concept in ecology and evolution, but they have not been good models for genetics. And that's what we are trying to do. We are trying to develop this side to bring the genetics into a model where we have a good understanding of sexual conflict. So we need models where we understand sexual conflict. We need models where there's a lot of variation of these sexually antagonistic traits, so we can do some mapping and we can try to trace this genetically. And of course, this model has to be able to allow us to do some at least basic genetics. So for the first part, so water striders are known, and this is the reason why they have been models for sexual conflict. They are known to have this concept or these kind of patterns really widespread in water striders. So there are all kinds of phenotypes and traits. So here there are two examples. I'm going to mostly focus in this one here, where the males have these two outgrowths of the 7th abdominal segment called male abdominal processes, and I will show you what the role of these in a second. There's another species where, for example, this genital segment has been changed to have this really large spine, where actually it has a role in opening the genitalia of the female when the female is trying to escape. So male abdominal processes, you can see them here in blue, highlighted in blue. They target a structure at the tip of the genitalia of the female called the proctiger, which is this last segment here, hidden here. If you dissect it, it looks like this. And they basically sit really nicely on this proctiger, and it allows the males to resist the female and also to open the genitalia to copulate. So one of the first things we started to notice is we have another species. So this one called Geras odontogaster. So this is a widely distributed species in Europe. And this is called Geras bruinoi. It's a sister species that you only find in North America, mostly in southern, in Canada and the United States. So this one, males have these male abdominal processes, and this one they don't have. It's really a presence-absence situation. So in this one, the abdominal processes target the proctiger of the female, which has this shape. And what we noticed in Geras bruinoi is that the proctiger of the female is exposed, whereas in this species here, the proctiger is concealed, and it's mostly concealed because of this plate here, which is a modified 8 abdominal segment, which is quite large, as opposed to Geras bruinoi, which is narrower, leaving the proctiger exposed. So this gave us a hypothesis, and the hypothesis is that because males target the proctiger of the female, so that probably puts selection of the female to conceal their target, to conceal the proctiger, and that would be a case of escalation or a case of correlated evolution between males and females. So to do this, we made a bunch of predictions. So then if there is variation in this trait in the population, then there should be also variation in the female. So we found this paper here from the 70s where they describe the population in Italy where the males have a rudimentary proctiger, a rudimentary process, which is a population of the same species, Geras odontogaster. So what we predicted is that because they have small proctiger, probably non-functional proctiger, the females will have evolved accordingly, and the shape of the process and the shape of the proctiger of the female will be probably exposed. So we took the car, we went to Italy, and we found actually that species. And to our amazement, if you compare the picture, this is from 2022 and this is from 1979, you could see that it's almost identical. It's really precise. And to our also big delight, the proctiger of the female of this population is quite exposed if you compare it to this. So we sampled a few more populations, and we did a correlation between male trait and female trait. So this is the female trait here on this axis, this is the male trait here, and you can see that this correlation holds in a very strong way. So this shows that it's most likely a pattern of correlated evolution between males and females. There is another prediction also that would test this hypothesis, and the prediction is that if a change in a population in the male, if the males of a population where they have the trait are put with the females of the population where they are not used to this trait, the males of the first population should have much easier time to mate with female from the other population than from females of their own population, because presumably females from their own population have already adapted to them and they know how to reject them. So we tested that, and we did it by testing performance in these mating trials. And what we did, we took males of Geras odontogaster with the trait, and we put them with females from Italy which have exposed proctiger. So the prediction is that these guys should have easier time mating with these females and more than what they would with their own females. And that's what you see in this graph here. So this prediction is also confirmed, and this further gives evidence that the traits in males and females are correlated in their evolution. So this gives us now our model, which is we have two species with presence-absence of traits where males and females are evolving in a correlated way, and now we can actually start to go and ask what are the loci underlying male trait, what are the loci underlying female trait, and how do these loci dynamically evolve through time. How much time do I have? 2? Okay. Can I have 3 more minutes? 2. Negotiation. Okay, so what we did, we built genomes, and that's the first thing we need. We looked also at how the traits develop during development. We tried to figure out the timing, and the timing is always at the end of development. That's where they start to appear. So these two eyes are basically developing processes of the males, and then you can stain them with all kinds of stainings. We crossed these animals, and just the first thing I want to show you is one of the main predictions in the field is that most of these sexually antagonistic loci will accumulate in the X chromosome. And that is not at all what we see in our system. But this is also something that others didn't see in their system. So if you cross Bruinoi with Odontogaster male with female, so these are the parental lines here, and these are the F1 males. So these males are heterozygous for all autosomes, but one of them has X chromosome of Odontogaster and the other one has X chromosome of Bruinoi, and you can see that they overlap almost perfectly. And what this means is that most likely, if there are any contribution from the X chromosome, it's going to be very small, and probably everything is on the autosomes. With females, it's different because they are now heterozygous for all chromosomes, including the sex chromosome, and we see that there is some maternal effect here. So this probably going to help us know at least what to predict in terms of mapping. We crossed this for many generations, and basically by doing that we are reshuffling the genome of these two. So these are again the parental lines, and you can see the 10th generation here, and you can see all the variation has been recovered, and these are some examples of this. We did some association mapping with this, and I'm going to make a long story short. Basically, we found about 200 SNPs that are associated with about 400 genes, which we filtered in all kinds of ways using comparative transcriptomics and other data sets. And we tried to figure out which genes are there. So the first observation is there are no hogs between those genes that harbor SNPs. So the abdominal B I showed you that is involved during the development is most likely not the gene where selection is acting to do this. I'm just going to show you one example, and this is the example of the gene notch where we found the SNP, which is right after the first exons. And when we knock down notch with RNAi, we get a phenotype like this. So this is a control, and you can see the abdominal processes. This is a late RNAi, and you can see we lose the processes. The animals are naked because notch is a neurogenic gene, so they have no bristles. And if we inject a little earlier, it's a much more dramatic phenotype. So this is still very preliminary. We are still screening all the candidates and also trying to enforce this data. But this is just to tell you that we have been really building this model to try to get this correlated evolution and escalation between the sexes. And now we are set to start now to look at male traits and female traits and what loci are involved and how do they evolve in a dynamic way during evolution. So I'll stop here. I just would like to thank the people who contributed and you for your attention. And if there's any time, I'll take any questions.